హలో అండ్ వెల్కమ్ టు Sreenath అకాడమీ. ఈ వీడియోలో మనం ఇంటర్మీడియట్ ఫస్ట్ ఇయర్ బాటనీ యొక్క చాప్టర్ 10 బయోమాలిక్యూల్స్ గురించి పూర్తిగా తెలుసుకుందాం. Hello and welcome to Sreenath Academy. Today we are going to discuss about the first year botany. So that is about the unit 5 chapter 10, that is about the biomolecules. So దీంట్లో we are going to discuss about the various topics under this particular chapter. So first one వచ్చేసి it's about the how to analyze the chemical composition. Second one is about the primary and the secondary metabolites. మూడోది వచ్చేసి about the biomolecules. నాలుగోది వచ్చేసి about the proteins. అండ్ ఐదోది వచ్చేసి పాలీసాకరైడ్స్. సిక్స్త్ వన్ వచ్చేసి న్యూక్లిక్ యాసిడ్. సెవెంత్ వన్ ఈస్ అబౌట్ ద స్ట్రక్చర్ ఆఫ్ ద ప్రోటీన్స్. అండ్ ఎయిత్ వన్ ఈస్ అబౌట్ నేచర్ ఆఫ్ బాండింగ్ లింక్ బిట్వీన్ ద మోనోమర్స్ ఇన్ ద పాలిమర్. అండ్ కమింగ్ టు ద నైన్త్ వన్ ఇట్స్ అబౌట్ ద డైనమిక్ స్టేట్ ఆఫ్ బాడీ కాన్స్టిట్యూయెంట్స్. అండ్ టెన్త్ వన్ ఈస్ అబౌట్ ద మెటబాలిక్ బేసెస్ ఆఫ్ ద లివింగ్. అండ్ 11th వన్ ఈస్ అబౌట్ ద లివింగ్ స్టేట్స్. So coming to the 10th chapter, biomolecules of 5th unit, that is the cell. So here we are describing about the introduction to the different kind of biomolecules, how the molecules are generally, what is the molecules which are making the living organism, that is the cells of the organism. Hello and welcome to Sreenath Academy. Today we are going to discuss about the unit 5, that is the chapter 10 biomolecules. So ఈ biomolecules లో మనం మాట్లాడుకునే the introduction లో we are going to discuss some of the molecules which are having some of the basic ingredients and making the basis of the life. So biomolecules అనగా ఏంటంటే they are the one which generally make up the cell, that is generally they will make up the cell and also they make up the function of the cell. So ఈ బయోమాలిక్యూల్స్ అన్నప్పుడు మనకు గుర్తు రావాల్సింది ఏంటంటే జనరల్గా ఏ కాంపోనెంట్ అయితే మనకు కార్బన్ ఉంటుందో, సో ఏ కాంపోనెంట్లో అయితే మనకు కార్బన్ ఉంటుందో ఆ కార్బన్తో పాటు హైడ్రోజన్, ఆక్సిజన్ ఇవన్నీ కలిసి ఉన్న దాన్ని మనము ఆర్గానిక్ కాంపౌండ్ అంటాము. సో దీస్ ఆర్గానిక్ మాలిక్యూల్స్ విచ్ ఆర్ ప్లేయింగ్ ఏ మేజర్ రోల్ in making the entire structure of the cell and also the function of the cell are called as biomolecules. So the organic components are the one which are generally making the entire biomolecules, and these biomolecules are coordinated and constructed together, which generally leads to the formation of a entire integrity of the cells and the structure, which is finally showing the function, that is the functions of the biomolecules. So here we can see the introduction to biomolecules law. We are going to discuss some of the important things which are generally discussed, which are making the basis for the life. And thereafter we are going with the first chapter, that is how to analyze chemical composition. So here we can see what are the different components which are generally chemically analyzed by means of different kind of structures. So what are the chemicals which are generally used to analyze the molecules or analyze the substances. So in the first topic we are going to discuss how to analyze the chemical composition. So if you are trying to understand what is the living form and what is non-living form, and what is the composition of the living form and what is the composition of the non-living form, so there are different kind of technology and the machineries which are generally involved in maintaining or trying to get the data of the materials and ingredients which are making the both the living and the non-living matter. So దీంట్లో చూసుకుంటే మనకు for example, if here in this kind of comparison of the elements between the living and non-living, so here we can see these are the different inorganic ions and that is the inorganic molecules which are generally making the earth crust, and also which are generally covered with the percentage of weight in case of human, that is the living matter. So here we can see the comparison of the elements which are different and very abundant in the nature, which are generally forming as a percentage of weight for a earth crust in comparison to that of the human being, that is the human body. So we can see the carbon. So the carbon is generally making about 0.03%. Here in the human body it is making 18.5%. So the percentage of that is the percentage weight of the carbon is very high in case of the human body when compared to the earth crust. So this is about the comparison between the both the living and the non-living matter. And coming to the they are different kinds. So apart from this tabular column we also discuss about the representation of the inorganic constituents. So how the electrolytes especially or the minerals are generally present within the formula. So here we can see that these are the formulas which are generally showing about the these are the most abundant and frequently observed inorganic constituent or inorganic inorganic ions which are making the living tissue. So here we can see the sodium is represented by Na, potassium is K plus, calcium is Ca plus 2, magnesium is Ca plus 2. So these three, this is the monovalent. So these are called as monovalent cations. These are called as divalent cations because they can able to form two bonds, and this is the monovalent because they can able to form the single bond. Ide gaakunda we also come across the water, NaCl, CO, CaCO3, phosphate and sulfate. So these are the different molecules which are also making up, that is the living matter, that is the inorganic composition or the constituents of the living matter. So this is the tabular column which is generally showing the comparison of the element between the living and non-living, as well as about the representation of the representation of inorganic constituents or the components which are generally making the living tissue. So next వచ్చేసి మనం మాట్లాడుకునేది ఇదంతా ఒక ఎత్తు అయితే, మనం ఒకవేళ ఒక సెల్ తీసుకొని, సో ఒక సెల్ యొక్క మనము దాని యొక్క ఆర్గానిక్ కాంపోజిషన్ అంటే నథింగ్ బట్ ద మాలిక్యూల్ విచ్ ఆర్ కంటైనింగ్ ద కార్బన్. ఇఫ్ యూ వాంట్ టు నో అవుట్, సో జనరల్గా దే విల్ టేక్ ద సెల్స్, దే విల్ బి సబ్జెక్టింగ్ టు ద సెంట్రిఫికేషన్. సో ఈ సెంట్రిఫికేషన్ సబ్జెక్షన్ చేసే ముందు దే ఆర్ యాడింగ్ ద ఏ కాంపోనెంట్ కాల్డ్ as ట్రైక్లోరో, okay, the trichloroacetic acid. So in the trichloroacetic acid is the component which is generally added to the cell suspension, and when they are subjected to the centrifugation, we can say that we are getting two different fractions. One is the acid soluble fraction, another one is the acid insoluble fraction. So the acid soluble fraction is nothing but it is the filtrate, which is a component, or it is the component which is consisting of all the compounds which are acid soluble, whereas acid insoluble uskunte, acid insolubles are the one which are generally they won't come in the filtrate, generally filter gaakunda they will be present within the precipitate. So here, because of after subjecting the centrifugation, we can see the cells or the cells or the organic matter is generally subjugated to the acidification and followed by centrifugation, where we can see we can get two different components. One is the acid soluble and the acid insoluble. So acid soluble is a filtrate which is consisting of all the minor and the macro biomolecules present within the suspension form, whereas acid insolubles are the one which are present within the precipitation, and we can see that is generally forming the chemical composition of the cell. So ఇక్కడ చూసుకుంటే దాని తర్వాత నెక్స్ట్ వచ్చేసి it's we are going to discuss about some of the biomolecules. So in case of here we can see what are the biomolecules. So the biomolecules are nothing but they are the one which are they are the molecules which are generally making the life of the cell. So the life of the cell, and which are generally important for the maintaining the structure and also function of the cell, are called as biomolecules. So ఈ biomolecules ని మనం ఇంకో విధంగా కూడా పిలవచ్చు, that is biomacromolecules. So what are biomacromolecules? The biomacromolecules are nothing but these molecules are abundant in nature and they are macro in size, so they are called as biomacromolecules. So in case of biomacromolecules, we come across different kind of molecules, like it is generally the amino acids and the proteins which are one category. So amino acids, proteins are the one category, and next thereafter it is about the lipids, okay, and followed by the carbohydrates, that is the polymer of carbohydrates, and nucleic acid. Okay, these are the different kind of biomacromolecules which are generally making the cell. So the amino acids and the proteins are the first category, and next one is the lipids, carbohydrates, and the nucleic acids. Okay, these are the molecules which are generally forming the entire constituents of the cell, and thereafter the organism, and make up the organism or build up the organism. So first category of biomolecules which we are going to discuss, it's about the amino acids. So what are amino acids? The amino acids are nothing but they are the component which are having the alpha carbon linked to the hydrogen atom, ammonium ion, and also carboxylic ion. So these are generally connected to alpha carbon called as amino acids. So amino acids are the one which are generally consisting of all these groups which are connected to the alpha alpha carbon, and దీన్ని మనము amphoteric అని కూడా అనొచ్చు, because it is consisting of positive and negative charge present on single compound called as amphoteric in nature. ఇంకోటి ఏంటంటే మనం ఈ అమైనో యాసిడ్స్ ని మనం ఎన్నో విధాలుగా డివైడ్ చేసుకోవచ్చు based on the side chain. So what we are seeing here, it is basically the side chain. So simplest form of side chain is observed in case of glycine, whereas aniline లో చూసుకుంటే మనకు జనరల్ గా మిథైల్ గ్రూప్ అనేది ఉంటుంది, సిరైన్ లో మనకు మిథైల్ హైడ్రాక్సిల్ గ్రూప్ ఉంటుంది. ఇదే కాకుండా మనకు కొన్ని బేసిక్ అమైనో యాసిడ్స్ ఉంటాయి, కొన్ని అసిడిక్ అమైనో యాసిడ్స్ ఉంటాయి. ఫర్ ఎగ్జాంపుల్ బేసిక్ వచ్చేసి హిస్టిడిన్ కానీ లైసిన్ కానీ, అసిడిక్ అమైనో యాసిడ్స్ వచ్చేసి మనము వాటికి అసిడిక్ గ్రూప్ ఉండడం అంటే కార్బాక్సిలిక్ గ్రూప్ ఉండడం సైడ్ చైన్లో అనేది చూడవచ్చు అనమాట. సో మనం మాట్లాడుకున్నట్టు దీస్ ఆర్ ద వన్ విచ్ ఆర్ కమింగ్ అండర్ ద కేటగిరీ ఆఫ్ amphoteric in nature, that is they are having the both the that is they are generally consisting of both the charges on particular component or particular compound, and we can also say that the amino acids are the one which are the building blocks. So they are the blocks or they are the building blocks of proteins which are generally bound together is called as proteins. So each and every amino acids are generally bound by means of peptide bond, so that this group of amino acids will be arranged in a sequence and they can form a building block of the proteins. ఇంకోటి ఏంటంటే నెక్స్ట్ కాన్సెప్ట్ వచ్చేసి మనకు 2 మార్క్స్ లో చాలా ఇంపార్టెంట్, దట్ ఈస్ అబౌట్ వాట్ ఈస్ జ్విటర్ అయాన్. సో జ్విటర్ అయాన్ అంటే ఏంటంటే జ్విటర్ అయాన్ is nothing but it is the state of the amino acid where we can see the presence of both positive and negative charge together. అంటే చార్జ్ ప్రకారంగా చూసుకుంటే న్యూట్రల్ లెవెల్లో ఉంటే దాన్ని మనం జ్విటర్ అయాన్ అంటాము. సో జ్విటర్ అయాన్ is nothing but every amino acid will have their own జ్విటర్ అయాన్ pH. So zwitter ion is the status of the amino acid where we can see that is దాని యొక్క PI value is equal to the pH value. So PI value is nothing but it is the value of the amino acid that is generally shown by the amino acid of one particular figure anamata. So when the PI of that particular amino acid, if it is equal to the pH of the solution, so pH అనేది it's a variant of the solution, whereas PI is nothing but it is the charge shown by that particular amino acid at a particular particular acidity or basicity of the solution. So whatever the charges which are generally showing, those charges are the one which are generally called as that is they are having both positive and negative charge together, it is called as zwitter ion. So zwitter ion annappudu always they are having the charge that is net charge is equal to 0, that is neutral in nature. So ee neutrality enduku ostundante because it is having both positive and negative charge together which will be cancelled. So because of the cancellation of the positive and negative charge, they are generally having the negative charge of 0. అదే కాకుండా మనం అసిడిక్ అమినో యాసిడ్ అంటే అసిడిక్ pH లో చూసుకుంటే మోస్ట్ ఆఫ్ ద అమినో యాసిడ్స్ ఆర్ హ్యావింగ్ ద పాజిటివ్ చార్జ్. అదే మనం బేసిక్ pH లో చూసుకుంటే మోస్ట్ ఆఫ్ ద అమినో యాసిడ్స్ ఆర్ హ్యావింగ్ ద నెగిటివ్ చార్జ్. సో దిస్ టెండెన్సీ ఆఫ్ షోయింగ్ both the charge together on a single compound is called as zwitter ion. సో zwitter ion అన్నట్టు మనం pH is equal to PI వాల్యూ అన్నమాట. సో మనం మాట్లాడుకున్నట్టు అసిడిక్ నేచర్ అంటే అసిడిక్ ఎన్విరాన్మెంట్లో ఉన్నప్పుడు పాజిటివ్ ఛార్జ్ ఉంటుంది, బేసిక్ ఎన్విరాన్మెంట్లో ఉన్నప్పుడు నెగిటివ్ ఛార్జ్ అనేది మనకు అమినో యాసిడ్స్లో ఉంటుందన్నమాట. సో నెక్స్ట్ వచ్చేసి వీఆర్ డిస్కసింగ్ అబౌట్ ద నెక్స్ట్ బయోమాలిక్యూల్ దట్ ఈస్ అబౌట్ ద లిపిడ్స్. సో లిపిడ్స్ ఏంటంటే జనరల్గా మనకు కామన్ కైండ్ ఆఫ్ నార్మల్ లిపిడ్స్ వచ్చేసి దాన్ని మనం ఎస్టర్స్ ఆఫ్ ఫ్యాటీ యాసిడ్స్ అంటాం. సో ఈ ఎస్టర్స్ ఆఫ్ ఫ్యాటీ యాసిడ్స్ అన్నప్పుడు ఫ్యాటీ యాసిడ్స్ ఆర్ షోయింగ్ ద ఎస్టర్ లింకేజ్ విత్ ఏ ఆల్కహాలిక్ మాలిక్యూల్. సో ఇక్కడ వచ్చేసి ద ఆల్కహాలిక్ మాలిక్యూల్ వచ్చేసి గ్లిజరాల్ అనమాట. సో whatever the lipids we are discussing, so the lipids which we are discussing most probably they will belongs to a class called as triglycerides. So triglycerides are generally formed as a result of esterification of the alcohol with the carboxylic group containing fatty acids. So the carboxylic group containing fatty acids are forming the esterification or ester linkage with that of the alcohol of the glycerol. So this is generally leading to the formation of a component called as lipid lipid, which is in technical terms we can call based on the what is the fatty acids are present. So here they are called as triglycerides, where we can see 1, 2 and 3. So there where we can see the three fatty acids are generally linked to one molecule of glycerol. So this is about the triglycerides, where we can see R1, R2, R3 or the fatty acids which are linked to the glycerol. So మనము ఇక్కడ కొన్ని ఎగ్జాంపుల్ చూస్తున్నాం. ఫర్ ఎగ్జాంపుల్ వన్ ఆఫ్ ద మేజర్ అండ్ వన్ ఆఫ్ ద మోస్ట్ abundant one is called as lecithin, which is called as a phospholipid, which is present as the most abundant lipid within the lipid membrane, that is membrane of the cell. So అదే కాకుండా మనం నెక్స్ట్ వచ్చేసి we also discuss about the one of the most important one which is coming under the category of lipids and fats is nothing but the cholesterol. So cholesterol ante endante these are coming under the category of steroid nuclei. Ante వీటికి ఏముంటాయి అంటే జనరల్ గా they are consisting of a steroid nuclei with a alcoholic group. So they are having the alcoholic group linked to the steroid nuclei. So these molecules are called as cholesterols and they are coming under the category of fats and the lipids. Inkoti endante one of the most important manaku nucleic biomolecule వచ్చేసి that is about the nucleic acid. So nucleic acids అంటే ఏంది జనరల్ గా మనం ఎట్లా ప్రోటీన్స్ ఆర్ ద పాలిమర్స్ అంటామో న్యూక్లిక్ యాసిడ్ కూడా పాలిమరిక్ మాలిక్యూల్ అన్నమాట. సో ఈ పాలిమరిక్ మాలిక్యూల్ దేంతో ప్రిపేర్ అయి ఉంటాయి, దేంతో మేడప్ అయి ఉంటాయి అంటే బికాస్ ఆఫ్ ద మోనోమెరిక్ ఫార్మ్. సో వాట్ ఆర్ ద మోనోమెరిక్ ఫార్మ్ హియర్? దే ఆర్ ద న్యూక్లియోటైడ్స్. సో ద న్యూక్లియోటైడ్స్ ఆర్ ద మోనోమెరిక్ ఫార్మ్ which is generally joined together to form a polymer, that is the nucleic acid. So ఈ పర్టికులర్ న్యూక్లియోటైడ్స్ ఏవైతున్నాయో ఈ న్యూక్లియోటైడ్స్ ఆర్ జనరల్లీ మేడప్ ఆఫ్ డిఫరెంట్ ఇంగ్రీడియంట్స్ అన్నమాట. సో ఇక్కడ డిఫరెంట్ ఇంగ్రీడియంట్స్ వచ్చేసి ఏంది మనకు ఫస్ట్ వచ్చేసి మనకు నైట్రోజన్ బేస్, రెండోది వచ్చేసి ఇట్స్ అబౌట్ ద షుగర్ అండ్ మూడోది వచ్చేసి ఫాస్ఫేట్. సో మనకు ఏంది మనకు ఈ మూడు కలిపితే ఒక్క న్యూక్లియోటైడ్ ఫామ్ అవుతుంది. అంటే మనకు ఒక్క న్యూక్లియోటైడ్ ఫామ్ కావాలంటే మనకు నైట్రోజన్ బేస్, sugar and phosphate, they should combine together to form one nucleotide, and this one nucleotide is generally polymerized to form a polymer. So we can see that generally here we can see that the adenine and the uracil, these are nothing but adenine వచ్చేసి it's a two member ring which is a heterocyclic component, and whereas the uracil is the pyrimidine which is a one member molecule. So ఈ విధంగా మనము మాట్లాడుకున్నట్టు ఈ విధంగా మనం ఈ నైట్రోజన్ బేసెస్ ఫస్ట్ దే విల్ కంబైన్ విత్ ద నైట్రోజన్ బేసెస్ విల్ కంబైన్ విత్ ద షుగర్స్ అన్నమాట. సో దిస్ ఈస్ ద షుగర్ మాలిక్యూల్. సో ఆ విధంగా ఫామ్ అయిన దాన్ని మనం ఏమంటాం? న్యూక్లియోసైడ్ అంటాం. సో ఫైనల్గా న్యూక్లియోసైడ్కి ఏమవుతుంది? ఫాస్ఫేట్ వచ్చి బైండ్ అవుతుంది. సో మీరు ఇక్కడ గుర్తుపెట్టుకోవాల్సింది ఏంటంటే సింపుల్గా మనకు ఫస్ట్ నైట్రోజన్ బేసెస్ ఉంటాయి, ఐదర్ ఇట్ మే బి ప్యూరిన్స్ ఆర్ పిరిమిడిన్స్. ఈ నైట్రోజన్ బేసెస్ వచ్చి దే విల్ బైండ్ ఆన్ టు ద షుగర్ మాలిక్యూల్. Okay, they will bind onto the sugar molecule to form the nucleotides. ఆ విధంగా ఫామ్ అయిన న్యూక్లియోటైడ్స్ ఏవైతున్నాయో they will be having the phosphate molecule or phosphate moiety which is generally bound with the nucleoside to form the nucleotide. So ఈ విధంగా ఫామ్ అయిన న్యూక్లియోటైడ్స్ ఏవైతున్నాయో they will be arranged on the each other. So ఒక nucleotide will be stacked upon the other nucleotide and the other nucleotide will be stacked upon each other. So this kind of stacking is nothing but it is because of the presence of a bond called as phosphodiester bond. So what is phosphodiester bond? Phosphodiester bond is the one which is generally a linkage between the two individual monomeric nucleotides. So the two individual monomeric nucleotides are joined together by means of a phosphodiester bond. So here we can see that the lipids are the one which are generally we can say that they are having the glycerol and fatty acids which are combined together, and also we can see that the nucleotides are also consisting of phosphate, sugar and nitrogen bases, and we can also say that generally the nucleic acid is also a polymer which is consisting of a monomeric form of nucleotides. So in the second kind of topic we are going to discuss about the primary and secondary metabolites. So in case of what are the primary metabolites, what are the secondary metabolites are generally observed in this particular paragraph, and we can see that the primary metabolites are the one which are useful for the daily life of the plant, whereas secondary metabolites are the one which are generally formed as a result of different kind of a complex metabolic pathway, where we can see these are the list of secondary metabolites metabolites which are generally cannot be used by the organism, but it's having the economical importance. So coming to the second topic, that is about the primary and secondary metabolites. So what is primary and what is secondary metabolites? Metabolites ante these can be the end products of the reaction, or they can be the intermediates of the reaction, which is nothing but called as metabolic reaction. So the organic reactions which are taking place within the cell is called as metabolites or metabolic reactions. So the metabolic reactions are the one which can be called as primary and the secondary metabolites. So primary metabolites are nothing but they are the one which are very much essential. So they are essential for the growth of the organism. Okay, whatever the things which are generally prepared by the organism and very essential for the day-to-day life, for all the cellular activities and functions of the cell is called as primary metabolites. So primary metabolites and they are very important for the daily functions of the cell. So it can be the proteins. Okay, it can be, for example, it can be proteins, that too in the form of enzymatic form. So they can be the enzymes or the proteins, and also they can be the carbohydrates. Okay, they can be the enzymes or the proteins and the carbohydrates, or any kind of molecule which are very much essential for the functioning of the cell. Ade manam secondary metabolites ante endante general ga they are the one which are leastly involved in case of metabolism of the plant, but most of the time they are generally used for the defense and the attack mechanism. So the secondary metabolites are the one which are polymeric in form, and they are not useful for the cellular metabolism, but they are used by the organism in a defense mechanism. So these are the list of the secondary metabolites. So secondary metabolites are the one which are generally formed as a result of polymeric or polymerization of the components. So for example, we can see the carotenoids or xanthophylls and also anthocyanins. ఇవే కాకుండా మనము ఆల్కలాయిడ్స్ అంటే మార్ఫిన్, కాల్సిడిన్. సో these are the some of the, they have the medicinal values, and also they are used as a drug of abuse. Ade kaakunda we can also see that the essential oils like lemongrass oil and toxins like resin and the drugs that is the drugs which are generally anti-cancer in condition, that is vinblastine is an anti-cancer drug. Ide kaakunda we also think that generally the rubber, gums and cellulose are the one which are the polymeric substances. So here these are the primary and secondary metabolites which are generally present, and they are the one which are important for the cellular activity. So primary are the one which are used by the cellular activity, and secondary is the one which is generally called as which are generally utilized by the plant, or it can be called as like. So secondary metabolites are generally used in case of defense and the all kind of other activities. So third one is biomacromolecules. So the biomacromolecules here we can see ఇక్కడ ఏంటంటే జనరల్గా మనకు చాలా మటుకు ప్రోటీన్స్ కానీ, అమినో యాసిడ్స్, న్యూక్లిక్ యాసిడ్స్, కార్బోహైడ్రేట్స్ అండ్ ఆల్సో ఎవ్రీథింగ్ వాట్ ఎవర్ ద థింగ్స్ which are generally making the cells, those are called as biomacromolecules, which were described in the third topic. So coming to third topic, that is biomacromolecules or macromolecules. So here we can see these are the molecules which are generally having the molecular weight somewhere around 18 to 800 daltons, and they are generally made up of some of the basic ingredients like carbon, hydrogen, oxygen, and to some extent nitrogen and sulfur. So these are the elemental forms which are generally gathered together to form a macromolecule, that is biomacromolecule, and they are having the molecular weight between 18 to 800 daltons, and we can see that these are generally this is the average composition of the cells, or different kind of how the different kind of cellular masses are generally making the total cellular mass. So we can see the water, proteins are the one which are making 10 to 15%, carbohydrates 3%, lipids 2%, and nucleic acid 7 to 1%, ions 1%. So we can see the generally this is the percentage of the each and every component present within the cell according to the total total cellular mass. So according to the percentage of total cellular mass, these are the different components which are generally making this particular composition. So ఇక్కడ చూసుకున్నట్టు ఈ macro biomolecules ఏంటంటే they are the one which are larger in size when compared to the either it may be the naturally occurring minerals కానీ inorganic ions కానీ minerals కానీ ఇవన్నీ ఏంటంటే they are the naturally occurring molecules which are coming under the category biomolecules. So whatever these the biomacromolecules or they generally the biomacromolecules are the one which are generally molecular in size, that is the molecular size is macro in size, and they are much bigger, that is so when compared to the size, in case of size or when you are discussing about the size, they are generally present within the macro state. So they are called as macro biomolecules. So under this category we are going to discuss, as we said, about the proteins, nucleic acid, carbohydrates, that is polymer of carbohydrates, especially the starch and the glycogen, and these are the different molecules which we are going to discuss in detail. So all these molecules are nothing but called as macro biomolecules, which are generally consisting about the cells. So fourth one it's about the proteins. So here we can see in case of proteins, they are the polypeptide chains, nothing but called as a linkage between the monomeric form of amino acids. So coming to the fourth topic, that is about the proteins. The proteins are the one which are also called as macromolecules. These macromolecules are generally called as polymeric. So why they are called as polymeric? Because they are consisting of monomeric units joined together. So the proteins are the polymeric forms, but these polymeric forms has been constructed because of the joining of the monomeric form. So what are the monomeric forms here? The monomeric forms are nothing but the individual amino acids which are joined together or called as monomeric forms. So the monomeric forms are joined together as individually to create a bond by means of a bond called as peptide bonds. So if you are seeing here, this is the first amino acid, this is the second one, and third one, so on. Many amino acids are joined together to form a polymeric form, that is the proteins, and the bond between these particular amino acids are called as peptide bonds. So what are peptide bonds? The peptide bonds are nothing but these are the linking bonds between the first and second amino acid and so on to the end of the all the amino acids are generally joined together to form a polymeric form. So this bond is a covalent bond, and it is generally it is also resistant to the denaturation forces like heat and the other factors. So the peptide bond is the one which is a covalent bond formed between the amino acids, and they are the one which are joined together. The entire amino acids are joined together to form a polymeric form called as proteins. So ఈ proteins యొక్క biological function చూసుకుంటే, సో ఇప్పటివరకు మనం మాట్లాడుకుందంతా హౌ ద ప్రోటీన్స్ ఆర్ క్రియేటెడ్. సో దీని యొక్క బయాలజికల్ ఫంక్షన్ చూసుకుంటే మనకు దాని యొక్క స్ట్రక్చర్ ఈస్ ఆల్వేస్ రిలేటెడ్ విత్ ద ఫంక్షన్ ఆఫ్ ద ప్రోటీన్. సో ద ఫంక్షన్ ఆఫ్ ద ప్రోటీన్ ఈస్ ఆల్వేస్ కోరిలేటెడ్ విత్ ద స్ట్రక్చర్ ఆఫ్ ద ప్రోటీన్. దీన్ని అర్థం చేసుకోవాల్సింది ఏంటంటే మనకు స్ట్రక్చరల్ ప్రకారంగా రెండు రకాల ప్రోటీన్స్ ఉంటాయి. ఒకటి ఫైబ్రస్ ప్రోటీన్, ఇంకోటి గ్లోబ్యులర్ ప్రోటీన్. సో ఫైబ్రస్ ప్రోటీన్స్ ఆల్వేస్ ప్లేస్ ఏ మేజర్ రోల్ ఇన్ కేస్ ఆఫ్ constructing the cells, whereas the globular proteins always plays a major role in performing the interior functions of the cell. So we are having two different proteins, that is the structural proteins and the that is generally the fibrous protein. Another one is the globular protein, that is the spherical proteins which are having different kind of functions. So for example, here the first one is the collagen, which is a protein molecule that is generally present within the extracellular space between the cells, and that is called as ground substance. So the collagen is the one which is providing the mechanical strength and the support, and it is making the that is a matrix within the cell, and it is playing a major role as a structural and also mechanical protein. Inkoti next వచ్చేసి trypsin. So trypsin is an globular protein. So it is generally generally the trypsin is an enzyme which is involved in enzymatic process, and the trypsin is a globular protein. ఆ విధంగా చూసుకుంటే మనకి ఇన్సులిన్ is also hormone which is a globular protein. Antibodies are also generally they come under the category of glycoproteins. సో ఇక్కడ మనం చూసుకుంటే యాంటీబాడీస్ ఏవైతున్నాయో which are very important for fighting against the infection, they are the glycoproteins, because they are special kind of proteins, because they are consisting of carbohydrate moiety attached with the protein. So these are the one which are also called as antibodies, which are involved in fighting against the infections. Next వచ్చేసి we are discussing about the receptor molecules. So the receptor molecules like sense, taste and hormone receptor are also generally the structural proteins which are generally either present within the cytosol or within the nucleus or on the surface of the membrane. So the receptors are the molecules which are generally involved in this activity, and they are also proteinaceous in nature. And coming to the next one, that is the transporter, that is the GLUT transporters, which is also plays a major role in transporting the glucose, and it is playing a major role in transporting the glucose across the membrane. And when you are coming to describe about the structure of the transporter protein, that is the membrane transport proteins, they are also made up of protein, and they are generally sometimes made up of protein. సో ఫిఫ్త్ టాపిక్ ఈస్ అబౌట్ ద పాలీసాకరైడ్స్. సో దీంట్లో ఏంటంటే మనం మోనోమెరిక్ సాకరైడ్ యూనిట్స్ ఏవైతున్నాయో దే ఆర్ జనరలీ జాయిన్ బై మీన్స్ ఆఫ్ గ్లైకోసిటిక్ బాండ్స్, వేర్ వీ క్యాన్ సీ ద మోనోమెరిక్ ఫార్మ్స్ ఆర్ జనరలీ జాయిన్ టు ఫార్మ్ ద polysaccharides which are generally involved in various kind of functions. Especially we can see the glycogen and also surface carbohydrates which are involved in the identification of the cells. So coming to the fifth topic, that is about the polysaccharides. So the polysaccharides are the one which are generally this is the polymeric form. So dani man polysaccharides antamu. So what is polysaccharides actually? This is the polymeric form of sugars. So if the sugars which are more than 9 in number, if they are joined together, and this kind of polymer is called as polymer of sugar is called as polysaccharides. Poly means many, saccharides are nothing but the sugars. Many sugars, if they are joined together, it is called as polymer of sugars. So a polymeric form of sugars are nothing but called as polysaccharides, where we can see the individual molecules are nothing but called as monomers. So what are monomers here? The monomers are nothing but the individual molecules which are nothing but the simply called as sugars. So the sugars are the one which are individual molecules nothing but called as monomers, which are joined together to form a polymeric form of sugars called as polysaccharides. So దీంట్లో చూసుకుంటే ఏంటంటే మనకు ఈ మోనోమెరిక్ మాలిక్యూల్స్ ఏవైతున్నాయో ఇక్కడ డయాగ్రామ్ లో చూస్తున్నట్టు, so these are the monomeric molecules which are generally shown in the diagram. We can see this is the linear form of the polysaccharide, and here we can see the branching form of the polysaccharide. So this is the line diagram, and this is the diagram which is generally representing the collagen molecule. So collagen is one of the polymer, but that is generally a storage form of polymer of sugars within the animal. So ఇక్కడ చూసుకుంటే ఏంటంటే మనకు in each and every individual monomeric sugar molecules are generally joined together by means of a bond called as glycosidic bond. So the glycosidic bond is the one So the glycosidic bond is the one which is generally involved in creating a linkage between the individual sugar molecules, and we can see there are different kind of glycosidic bonds. So initial ga manam starting lo chuskunte linear form is the one which is consisting of alpha 1,4 linkage, whereas the branching form, so the branching form is consisting of alpha 1,6 linkage. So there are difference in the linkage. So the difference in the linkage is the one which is causing the difference in the pattern of the molecule. So if it is a linear form, it is showing the alpha linkage between the 1 and 4 carbon. If it is a branch form, it is showing the linkage between the first and the 6th carbon. So the first and 6th carbon, if they are forming the linkage, so that is the branching form. If it is forming the linkage between the first and 4th carbon, then that is the linear form of linkage. So these are the polysaccharides which are nothing but the polymer of glucose, which are generally involved either in the form of either it may be formed in the form of starch in case of plants, okay, that is the storage form, whereas in animals it is generally glycogen. So the glycogen is nothing but it is a storage form of animal carbohydrates. So the animals are the one which are having the storage form of carbohydrate, that is in the form of glycogen. Plants are the one which are generally consisting of storage form of polysaccharides in that is generally called as starch. And when we discuss about the structural role, most probably the structural role of the polysaccharides are generally they can create, they have extraordinary role as a structural role. So here we can see, for example, the one of the best example is the that is the cellulose. So cellulose is a polymer. Cellulose is also a polymer, that is a polymer of sugars, which is generally forming the cell wall of the plant, that is the cellulic form of the plant, generally consisting of cellulose. Ide gaakunda we also discuss about they have a very eminent role in case of maintaining the glycoprotein cover of the RBC. So the glycoprotein cover of the RBC is generally made up of proteins and the carbohydrates together. So the glycoprotein cover is nothing but the proteins and the glycogen which are generally forming the cover on the cell, is the one which is generally providing the protection, and also it is providing some kind of charge, that is generally it is providing some kind of charge affinity present on the RBC, which is causing the RBC to move through the a minute form of capillaries. So ఈ విధంగా అర్థం చేసుకుంటే మనకి ఈజీగా అర్థమవుతుంది. సో పాలిమరిక్ ఫామ్ ఒకవేళ స్ట్రక్చరల్ రోల్ గురించి మాట్లాడుకోవాలనుకుంటే సెల్యులోస్ ఈజ్ వన్ ఆఫ్ ద ఎగ్జాంపుల్, అండ్ ద గ్లైకోప్రోటీన్ దట్ ఈస్ ద గ్లైకోప్రోటీన్ కవర్ that is present on the surface of the RBC is also one of the example. So sixth topic is about the nucleic acids. So in case of nucleic acids, we can see they are the DNA and RNA molecules are the one which are acting as a genetic material. So దీంట్లో మనం DNA యొక్క స్ట్రక్చర్, RNA యొక్క స్ట్రక్చర్ ఏంటి, and what are the different components which are making the polymer of nucleotides together is called as nucleic acids. So coming to the sixth topic, that is about the nucleic acid. So nucleic acid అనగా మనకు గుర్తు రావాల్సింది ఏంటంటే జనరల్గా it is a genetic material అన్నమాట. So it is the genetic material which is generally present within the cells. So for example, if you are seeing the higher organisms లో DNA, that is especially the double stranded DNA is the molecule which is generally making the genetic material of the higher organisms, whereas if you are seeing the lower organism, most probably it can be the single stranded DNA molecule, ade gaakunda double stranded RNA molecule. Okay, they are the different molecule which are generally acting as a genetic material in case of lower organisms. So higher organisms are the one which are generally consisting of double stranded DNA molecule. Lower organism is the one which is generally consisting of double stranded RNA, single stranded DNA, or maybe sometimes single stranded RNA molecule. So మనకు మెయిన్ గా దీని యొక్క మెయిన్ ఉద్దేశం ఏంటంటే జెనెటిక్ మెటీరియల్. సో దీన్ని ఎందుకు మనం జెనెటిక్ మెటీరియల్ అంటున్నామంటే బికాస్ ఇట్ ఈస్ హ్యావింగ్ ద టెండెన్సీ ఆఫ్ స్టోరింగ్ ద ఇన్ఫర్మేషన్. So the information which is needed for the activity of the cell has been stored, and also they are involved in the transmission. So ఎందుకు అంటే మనకు they are the one which are generally involved in storage of the material, that is the storage of the information is generally absorbed in case of nucleic acid, and also the transmission of the material is also absorbed in that is in the form of nucleic acid. So the nucleic acids are the one which are generally involved in storing the information and also passing off, that is the transmission of the information from one generation to the other generation. So ఈ nucleic acid ని మనం ఇంకో విధంగా కూడా మాట్లాడుకోవచ్చు, that is they are also polymers. ఏ విధంగా polymers? These nucleic acid, either it may be DNA or the RNA, they are the polymers. So they are the polymeric molecules or the polymers which are generally individually made up of what they are made up of. They are made up of monomeric forms. So what are the monomeric forms which are generally making the polymeric nucleic acid? These monomeric forms are nothing but called as nucleotides. So the nucleotides are nothing but they are the molecules which are joined together by the phosphodiester bond to form a polymeric molecule, that is the nucleic acid. So the nucleic acid is the one which is a polymeric molecule, and we can see the individual monomeric form, that is the nucleotides, are joined by means of phosphodiester bond to form a larger molecule, that is the nucleic acid. So especially in case of higher organism, we can see the double stranded DNA molecule as a genetic material which is generally stored and also has the ability to transfer the information from one generation to other generation, and it is generally present within the nucleus as a genetic material. మెటీరియల్. ఇంకోటి ఏంటంటే it also interacts with number of proteins for the purpose of folding. So we can see that the DNA material is very long and lengthy, but in order to show the folding and integrity properly, it will combine with the histone proteins, and sometimes it also work with the non-histone proteins like DNA polymerase to undergo replication process. So nucleic acids are nothing but they are very important for the life of the cell, because they are the commanding center of the cell, where we can see all the information has been stored, all the information has been passed on, and has been generally involved in this particular process of storing and transmission of the information. And we can see this genetic material is generally present, and they can be passed on to the future generation. So in case of seventh topic, that is about the structure of proteins. So in case of structure of proteins, where we can see that primary structure, secondary structure and tertiary structures are the one which are generally making the protein molecules. So in case of primary structure, we can see the peptide bond is the one which is linking between the different monomeric form of amino acids within the protein, and we can see these are the structures which are forming the secondary structure, that is the alpha helix, and third one is about the tertiary structure, which are generally forming the entire protein structure. And most of the proteins which are generally structural proteins and the enzymatic protein, that is the globular proteins, are generally consisting of tertiary structure. So coming to the seventh topic, that is about the structure of the protein. So proteins are also polymer of amino acids. So the amino acids are generally joined by means of peptide linkage or the peptide bond, where we can see different amino acids are joined together to form the protein molecule. So here the first kind of molecule, that is a polymer, which is generally formed as a result of joining of this particular amino acids, is the primary structure. So the primary structure is nothing but it is the first one which is generally formed as a result of formation of the peptide bond between the amino acids to form a linear molecule. So this is the hypothetical molecule which is generally present, where we can see the amino acids are generally joined together by means of peptide bonds, and we can say that the first amino acids having the free end, that is the ammonium end is free, whereas the last amino acid is having the carboxylic group as the free. So we can see all the amino acids which are present in between, they are having the engagement of both the nitrogen group, that is the amine group, and also carboxylic group are engaged together to form the peptide bond or linkage. So the peptide bond or linkage is generally formed as a result of between the different amino acids. We can see the amino acids are generally joined or started joining from the N-terminal end and finally end with the C-terminal end. So ఇక్కడ చూస్తున్న ఈ particular protein structure ఏదైతుందో, that is the linear form of polymeric molecule which is just formed as a result of formation of the peptide linkage between the amino acids, and this linear form of molecule is called as primary structure, which is the hypothetical protein which is generally seen as a reference from the N to C. So N is the starting point and the C is the ending point. So here this particular segment, if you are taking and if you are observing this one, we can see that these are the amino acids which are joined together by means of this peptide bond. So this is the peptide bond which we are rounding in the second trip, that is the one which is the peptide bond which are joining the different amino acids together and forming a primary structure. So ee vidhanga form aina ee linear molecule edaitundo, ee linear molecule ni man primary structure of amino acid anochu. So ee primary structure of amino acid is called as linear molecule or the basic structure of the protein. So ee vidhanga linear molecule kasta it will undergo bending. So what it will takes place, there is a bending of the molecule, that is the bending of the linear molecule will takes place to form a structure called as helical structure. So the helical structure is nothing but that is the molecule resulted as a result of bending of the linear form. So this molecule is called as helical structure, where we can see the helical format has been observed. So this is the secondary structure of the protein called as helical structure, which is forming as a result of bending of the molecule, that is the bending of the primary structure of the protein in the helical format. So ee helical format ఏదైతుందో ఈ విధంగా మనం డయాగ్రామ్ లో చూసినట్టు it is taking the structure of a helix, that is generally a spring kind of appearance of the protein is called as helical structure, which is generally formed as a result of hydrogen bonds. So the bonds which are generally present at the background or the present generally in the background of the protein molecule is the hydrogen bond bond which are generally involved in causing the helical structure, that is the helical format of the protein. ఈ విధంగా ఫామ్ అయిన హెలికల్ స్ట్రక్చర్ కాస్త it will also further undergo further folding. So the further folding of the protein will also takes place, and this further folding of the secondary structure of the protein, it is generally taking place and leads to a formation of a structure called as tertiary structure. So the tertiary structure is the one which is generally created as a result of further bending of this helical format into a globular molecule or a much more folded fashion called as tertiary structure. So all the proteins are generally takes place, that is the enzymatic molecules or nothing but the enzymes, and also the functional proteins like albumin, globulin, hemoglobin. Every molecule which are generally performing, they generally most of the time they will take the tertiary structure, especially the myoglobin, albumin, globulin, and also all the enzymes which are generally involved in enzymatic reaction within the cells are generally taking the structure called as tertiary structure. So the tertiary structure is the most stable one, and it is most common in case of the some of the most common molecules like myoglobin, which is involved in carrying the oxygen to the tissue level, and also albumin and globulin, which are generally present within the blood plasma and maintain the osmoregulation. And also we can see all the enzymes which are generally intracellular enzymes, and to certain extent the hormones are generally taking the tertiary structure. So this is about the structure of the protein. When we are discussing about the structure of the protein, మనం గుర్తుపెట్టుకోవాల్సింది ఏంటంటే అన్ని మాలిక్యూల్స్ లా కాకుండా ప్రోటీన్ మాలిక్యూల్స్ లో మనకు ఒకసారి ప్రోటీన్ మాలిక్యూల్ పాలిమర్ ఫామ్ అయినాక, అంటే ప్రోటీన్ పాలిమర్ ఫామ్ అయినాక కూడా మనకు దాన్ని అది కాస్త మనకు బెండింగ్ అండ్ ఆల్సో అది కాస్త మనకు ఫోల్డింగ్ అండ్ ఫర్దర్ బెండింగ్ జరగడం వల్ల ఆ పర్టికులర్ పాలిమర్ కాస్త ప్రైమరీ ఫామ్ ఆఫ్ ప్రోటీన్ ఉన్నది కాస్త మనకు బెండింగ్ అవ్వడం వల్ల సెకండరీ ఫామ్ నుంచి టెరిషరీ ఫామ్ కి కూడా ఫైనల్ గా అల్టిమేట్ గా క్వార్డినరీ ఫామ్ కూడా ఫోల్డ్ అయ్యే ఛాన్స్ ఉంటుందన్నమాట. సో మనకు అంటే జనరల్ గా మనకు కాంప్లెక్సిటీ పెరుగుతున్న కొద్దీ మనకు ఫోల్డింగ్ పెరుగుతుంటది, అండ్ లెవెల్ ఆఫ్ ఫోల్డింగ్ పెరగడం కొద్దీ మనకు లెవెల్ ఆఫ్ ఆర్గనైజేషన్ అనేది కూడా పెరుగుతుంటది. సో ప్రైమరీ ఈజ్ ద బేసిక్ వన్ విచ్ ఇస్ జనరల్లీ షోన్ యాజ్ ఎ రిజల్ట్ యాజ్ సూన్ యాజ్ ఇట్ ఈస్ సింథసైజ్డ్ ఇన్ ద నేసెంట్ ఫామ్. తర్వాత మోస్ట్ ఆఫ్ ద ప్రోటీన్స్ ఆర్ జనరల్లీ షో ద tertiary structure and the secondary structure. To certain extent they can also show the quaternary structure. So even the quaternary structure అయిన fourth level of organization ఉన్నా కూడా దాంట్లో కొన్ని segments will be having the secondary structure. So secondary structures ఉన్న proteins ని further గా fold చేయడం వల్ల ఒక complete globular molecule అయితే tertiary, ఆ విధంగా మనకు 2-3 molecules of tertiary folded protein ని ఒక దగ్గర కంబైన్ చేయడం వల్ల మనకు వచ్చేది quaternary అన్నమాట. So quaternary protein is the one which is carrying the tertiary protein. Each tertiary protein is generally consisting of helical format also, and also beta plated sheet together. So eighth one is about the nature of the bond which is linking the monomers in a polysaccharides or the polymers. So here we are describing about the nucleic acid that is generally seen here in case of the double stranded DNA molecule. So the links between the double stranded DNA molecules, here we can see that is the secondary structure of the DNA, where we can see the phosphodiester bond is the one which is linking the monomeric form of the nucleotides. So coming to the eighth topic, that is about nature of bonding linkage between the monomers within the polymer. So the polymeric molecule which is generally consisting of monomeric molecules which are joined together by means of bonds. So ikkada manamu this polymeric molecule which is generally showing the tertiary structure, that is the secondary structure of the DNA molecule. So manaku ikkada manam ardham chesukovalsindi endante manaku primary structure of the DNA molecule is nothing but a single stranded DNA molecule. Tertiary structure is nothing but that is the molecule which is showing the double stranded DNA molecule. So while discussing about what is the nature of the bond that is linking the monomeric forms within the polymer of DNA molecule, that is the phosphodiester bond. So the nature of the bond which is generally preparing or generally linking the monomers within the polymer is the phosphodiester bond. So the phosphodiester bond is the one which is generally present as a linkage between the monomeric form to form the polymeric molecule. So ఇక్కడ చూసుకుంటే మనకు మనకి ఇక్కడ చూసుకుంటే మనకు డబుల్ స్టాండర్డ్ డిఎన్ఏ మాలిక్యూల్ ఏదైతుందో దట్ ఈస్ నథింగ్ బట్ కాల్డ్ as secondary structure of the DNA. సో ఇక్కడ చూసుకుంటే మనకు ఈ విధంగా మనకు దిస్ ఈస్ ద వన్ న్యూక్లియోటైడ్ విచ్ ఇస్ అ థైమిన్ అండ్ దిస్ ఇస్ ద నెక్స్ట్ న్యూక్లియోటైడ్. సో ఇక్కడ మనం అర్థం చేసుకోవాల్సింది ఏంటంటే ఒక న్యూక్లియోటైడ్ వన్ స్టాండ్ ఈస్ కంటైనింగ్ వన్ సీక్వెన్స్ ఆఫ్ న్యూక్లియోటైడ్, అదర్ స్టాండ్ ఈస్ కంటైనింగ్ అదర్ సీక్వెన్స్ ఆఫ్ న్యూక్లియోటైడ్. సో ఇప్పుడు ఫర్ ఎగ్జాంపుల్ ఇది మనము ఇది ఒక మాలిక్యూల్ అనుకుంటే, ఇది థైమిన్ అనుకుంటే, ఇట్ ఆల్వేస్ పేర్ విత్ ద adenine. So always the T will pair with the T, that is the T always pair with the A, that is adenine. ఇక్కడ ఇంకోటి ఏంటంటే guanine is always pairs with the C, that is the cytosine. సో మనము ఇక్కడ ఏంటంటే జనరల్గా మనకు డిఎన్ఏ మాలిక్యూల్ గురించి అర్థం చేసుకోవాలనుకుంటే మనకు డిఎన్ఏ మాలిక్యూల్లో ఆల్వేస్ ఒక స్టాండ్ 3' to 5' అయితే ఇంకో స్టాండ్ 5' to 3' రన్నింగ్ ఉంటుంది. అండ్ ఇక్కడ ఇంకోటి ఏంటంటే అర్థం చేసుకోవాల్సింది మనకు ఒక స్టాండ్లో థైమిన్ ఉంటే ఇంకో స్టాండ్లో అడినైన్ ఉంటుంది, కాంప్లిమెంటరీ బేస్ పేరింగ్ అంటారు దీన్ని. సో ఆల్వేస్ అడినైన్ విల్ బేస్ పేర్ విత్ ద థైమిన్ అండ్ whereas guanine is always base pairing with the cytosine. So cytosine always shows the bonding, that is the hydrogen bonding with that of the guanine, and the cytosine that is generally thymine is always base pairing with the adenine. So we can see that the A always base pairs with the T and the G always base pairs with the C. So this kind of bonds which are generally shown between the nucleotides is called as hydrogen bonds, and we can also say that the bond which is generally present. So ikkada chuskochu the bond which is generally present, it is involved in linking the sugar to the other sugar. So this is called as phosphodiester bond. So what bond it is called as phosphodiester bond. So the phosphodiester bond is the one which is generally involved in linking one nucleotide with that of the other nucleotide. So one nucleotide molecule joining with the other nucleotide molecule is because of the phosphodiester bond which is involved in linking both the nucleotides together. ఆ విధంగా మనకు n number of nucleotides are joined together to form a polymeric form of a DNA molecule. So the nature of bond that is generally involved in linking the monomeric forms within the polymer of DNA molecule is the phosphodiester bond. So ninth one is the one where we can see the concept of metabolism. So here we can see the turnover and also metabolism which is generally observed, and we can see what is metabolism and how this biochemical method or the biochemical process has been done within the cell. So coming to the concept of metabolism, this is also explained as in terms of dynamic state of body. So dynamic state of the body is nothing but the concept of metabolism, where we can see that the metabolism reactions which are taking place within the living cells or the living things or living beings are nothing but called as dynamic state. And ఇక్కడ చూసుకుంటే ఏంటంటే జనరల్గా మెటబాలిజం అనే రియాక్షన్ ఏంటంటే జనరల్గా మన కెటబాలిక్ అండ్ అనబాలిక్ రియాక్షన్స్ టుగెదర్ ఆర్ కాల్డ్ as metabolic reactions, and we can see they are nothing but the enzymatic process or nothing but the enzymes which are generally involved in this particular process, nothing but called as where we can see the conversion of substrate into the product. So the substrate by means of this enzymatic reaction it will be converted into the product, and we can see this kind of entire reaction is called as metabolic reactions, which is nothing but it is involved in the process of living of the ఇండివిజువల్. సో ఒక పర్సన్ కానీ ఒక ఇండివిజువల్ కానీ మనకు అది లివింగ్ స్టేట్లో ఉందంటే ఖచ్చితంగా అక్కడ మెటబాలిజం జరుగుతూ ఉండాలి. మెటబాలిజం ఈస్ నథింగ్ బట్ ఇట్స్ ఎ కెటబాలిక్ అండ్ ఆల్సో ఎనబాలిక్ రియాక్షన్ విచ్ ఆర్ అకరింగ్ టుగెదర్ ఈస్ కాల్డ్ ఎస్ మెటబాలిజం. అండ్ మనకి ఇంకోటి గుర్తుపెట్టుకోవాల్సింది ఏదైతే మనకు అమినో యాసిడ్స్ కానీ అంటే మనకు బయోమాలిక్యూల్స్ కానీ ఏ బయోమాలిక్యూల్స్ అయినా కూడా అమినో యాసిడ్స్, ప్రోటీన్స్, న్యూక్లిక్ యాసిడ్ ఇవి కానీ లేకపోతే ఎంజైమ్స్ కానీ అన్నిటికీ మనకు ఒక లైఫ్ టైం ఉంటుంది, హాఫ్ లైఫ్ ఉంటుంది. దాన్ని టర్న్ ఓవర్ అంటాం అనమాట. సో వాట్ ఈస్ టర్న్ ఓవర్? టర్న్ ఓవర్ ఈస్ నథింగ్ బట్ ఇట్స్ ఏ లైఫ్ లైన్ ఆఫ్ ఎ పర్టికులర్ మాలిక్యూల్, వెదర్ ఇట్స్ ఏ biomolecule or whether it's an enzyme. If they are working in a particular reaction, after certain period of time they will undergo degradation and they will be eliminated from the system. So always we have to see that the concept of metabolism is very dynamic in nature. Metabolism is the process where we can see all the life reactions taking place within individual, and we can see that the enzymes and the biomolecules are also consisting of turnovers, that is generally a lifetime or the lifeline of a particular molecule where we can see the degradation of the molecules will be occurring. So 10th one is about the metabolic basis for the living. So here we are describing about the catabolic and anabolic pathways. So catabolic ante oxidation, anabolic ante synthesis pathways గురించి మాట్లాడుకుంటాం. And also the role of the ATP, that is adenosine triphosphate role గురించి కూడా మాట్లాడుకుంటాం అన్నమాట. So coming to the 10th topic, that is about the metabolic basis for the living. So what is metabolic basis for the living? ఇక్కడ మనం అర్థం చేసుకోవాల్సింది ఏంటంటే మెటబాలిక్ రియాక్షన్స్ ఏ విధంగా దే బికేమ్స్ ఎ బేసిక్ ఫర్ ద లివింగ్ ఆఫ్ ద ఇండివిజువల్. సో ద మెటబాలిక్ రియాక్షన్స్ ఆర్ ఆఫ్ టూ టైప్స్, దట్ ఈస్ ద catabolic reaction and anabolic reaction. So the catabolic reactions are nothing but called as oxidation reaction, where we can see the breakdown of the component. So the breakdown of the component is called as catabolic reaction or oxidation reaction, which is one part of metabolism, whereas anabolic reaction is nothing but that is a synthetic component, where we can see the synthesis of component or the compound will be taking place. So that is called as anabolic reaction. So the catabolic and anabolic together is called as metabolic. So catabolic ni ఏ విధంగా అనొచ్చు, మనము దాని ఆక్సిడైజింగ్ రియాక్షన్ అనుకోవచ్చు, బ్రేకింగ్ డౌన్ రియాక్షన్ అనుకోవచ్చు, లేకపోతే మనకు జనరల్గా కాంపౌండ్ బ్రేక్ డౌన్ అని కూడా అనొచ్చు. సో దట్ ఈస్ ద catabolic రియాక్షన్. అనబాలిక్ రియాక్షన్లు ఏంటంటే మనకు ప్రికర్సర్స్ని వాడుకొని లేకపోతే ఇంటర్మీడియట్స్ని వాడుకొని ఒక మాలిక్యూల్ని సింథసైజ్ చేయడాన్ని దాన్ని అనబాలిక్ ప్రాసెస్ అంటాం. సో అనబాలిక్ ప్రాసెస్ ఇస్ నథింగ్ బట్ ద సింథటిక్ ప్రాసెస్ వేర్ వీ కెన్ సీ ద సింథసిస్ ఆఫ్ ద మాలిక్యూల్ యాజ్ ద ఫైనల్ ప్రోడక్ట్ ఫ్రమ్ ద ఇంటర్మీడియట్ ఆర్ ఫ్రమ్ ద బేసిక్స్ ఆఫ్ basics or the precursor molecules. So ఈ విధమైన రియాక్షన్స్ ని మనం nothing but call as metabolic reactions, which is in turn it is called as combination of both the catabolic and the anabolic reactions. So in case of metabolic reaction, we can see that the breakdown of the compound is generally involved in releasing the energy. So ఈ ఎనర్జీ ఏదైతే రిలీజ్ అవుతుందో, so the energy which is generally released as a result of the catabolic process or the oxidation process, that energy is conserved in the form of ATP molecule. So and also we can see in case of anabolic reactions, we can see the energy are generally utilized. So here in case of anabolic reactions, we can see that the ATP molecule is generally involved in providing the energy and for the anabolic or the సింథటిక్ మాలిక్యూల్. సో మనకి ఏంటంటే జనరల్గా గుర్తుపెట్టుకోవాల్సింది, బ్రేక్ డౌన్ అవుతున్నప్పుడు ఎనర్జీ రిలీజ్ అవుతుంది, అది ఏటీపీ రూపంలో దాయించుకోవడము. అదే మనకు సింథసిస్ జరిగేటప్పుడు మనకి ఎనర్జీ అవసరం పడుతుంది, ఆ ఎనర్జీ కూడా ఏటీపీ రూపంలోనే వాడుకోవడం జరుగుతుంది అనమాట. ఇదే కాకుండా మనకు బ్రేక్ డౌన్ జరిగేటప్పుడు మనకు ఒకవేళ ఆక్సిడేషన్ రిడక్షన్ రియాక్షన్ జరిగితే NAD+ వచ్చేసి ఇట్ విల్ కమ్ అండ్ యాక్సెప్ట్ ద ఎలక్ట్రాన్స్ అండ్ ద ప్రోటాన్స్. అదే ఒకవేళ మనకు సింథసిస్ జరిగేటప్పుడు మనకి ఏంటంటే NADP+. So NADP+ molecules ఏవైతున్నాయో they are the one which are generally involved in contributing the electrons and the protons for building the molecule. So మనం ఇక్కడ గుర్తుపెట్టుకోవాల్సింది, బ్రేక్ డౌన్ అయ్యేటప్పుడు ATP జనరేట్ అవుతుంది, ఎలక్ట్రాన్స్ ప్రోటాన్స్ లిబరేట్ అవుతాయి, దట్ ఈస్ టేకెన్ బై NAD+. అదే సింథసిస్ జరిగేటప్పుడు ATP యూటిలైజ్ అవుతుంది అండ్ ఆల్సో మనకు NADP+ ఏదైతుందో ఆ NADP+ కాస్త మనకు ఆ పర్టికులర్ కాంపోనెంట్కి అంటే సింథసిస్ కావాల్సిన electrons ni protons ni deliver చేస్తుంది అన్నమాట. So that is about the metabolic bases which are a basic for the living of the organism. So final one is about the living state. So here we can see always the living state is generally dynamic in nature, and we can see there this particular system. So especially living state గురించి చెప్పుకోవాలనుకుంటే they are the one which are nothing but called as non-equilibrium steady state. So the living state is always present in a non-equilibrium steady state position where we can see they are able to perform the work. So that is generally observed in case of the biomolecules, that is within the cell. So coming to the 11th topic, that is about the living state. So what is living state? So living state is the one which is generally which is having all the characters of living organism or living growth and all kind of characters. So living state is nothing but it is generally a state of the individual where it is showing different kind of characters which are relevant with the growth and the living conditions. So for example, ఇంకోటి గుర్తుపెట్టుకోవాల్సింది ఏంటంటే మనకి లివింగ్ స్టేట్ అన్నప్పుడు గుర్తు రావాల్సింది దట్ ఈస్ అబౌట్ ద హోమియోస్టాసిస్. సో what is homeostasis? That is the maintenance of the some of the components in the equilibrium level, that is maintaining certain components in the equilibrium level is nothing but called as homeostasis. So here we can see that is generally the concentration of the glucose in case of normal individual, it is 4.5 to 5.0 millimoles. So that is the concentration of the glucose in a particular individual. And coming to the hormones, generally they are 1 nanograms per, generally we can measure in the form of nanograms per ml. So we can see every component is generally present in a particular state, that is called as that is nothing but called as equilibrium state, or the homeostatic state, where we can see these component has to present in that particular range only. It should not deviate from that particular range. So this is the state which is called as a steady state, or nothing but called as homeostatic state, where we can see the equilibrium maintenance of the molecules within the cell. Ikkada living state ni chuskunte manamu, they are the one which are non equilibrium, and they are the steady state which are involved in performing the work. So ఇక్కడ ఏంటంటే మనకు లివింగ్ స్టేట్ ఇస్ ద వన్ విచ్ ఇస్ జనరలీ ఇన్వాల్వ్డ్, అండ్ వీ కెన్ సీ దట్ దే ఆర్ ద వన్ విచ్ ఆర్ జనరలీ ప్రజెంట్ ఇన్ ఏ నాన్ ఈక్వలిబ్రియం స్టేట్. అంటే దాని అర్థం ఏంటంటే ఆల్వేస్ ద ప్రోడక్ట్స్ ఆర్ నాట్ ఈక్వల్ టు ద సబ్స్టేట్, బట్ ఓవరాల్ ద కాన్సన్ట్రేషన్ ఆఫ్ దట్ పర్టికులర్ category of molecule will be maintained constant అనమాట. So they are non equilibrium and they are generally showing the steady state. What is steady state? Always there is a proper way of regulation of the molecules. The concentration of the product and the concentration of the reactants are always maintained in a proper way and in a steady state. So they are not equilibrium and they are generally in the steady state and always performing the function. So these kind of things, that is generally non-equilibrium, steady state, and ready to do the work or performance of the work is called as living state. So this is about the things where we have to remember that what is generally the called as living state and the steady state or homeostatic condition of the organism. Coming to the summary of this particular chapter 10 of the botany 1. So here we are going with the summary of this particular chapter. Deentlo man we are going to give the introduction to biomolecules. So what are biomolecules, what are the molecules, and how the biomolecules are different from the other molecules, why they are called as biomolecules, and why they are involved, what is their involvement, whether they are structure and the function of the cell. That is about the introduction to the biomolecules. And coming to how to analyze the chemical composition. So there are number of methods by which we can analyze different biomolecules. So what are biomolecules? Actually the biomolecules are the one which are involved in maintaining the structure and functional integrity of the cell, and also in turn about the organism, that is the living organism. And ఇక్కడ గుర్తుపెట్టుకోవాల్సింది ఏంటంటే మనకు దాన్ని కనుక్కోవడానికి, అంటే మనకు ఏవైతే బయోమాలిక్యూల్స్ అనుకుంటున్నామో దాంట్లో మేజర్గా మనము ఎలాంటి ఎలిమెంటల్ ఫార్మ్స్ ఉంటాయంటే మోస్ట్ ప్రాబబుల్లీ ద మేజర్ one వచ్చేసి కార్బన్, హైడ్రోజన్, దాని తర్వాత నైట్రోజన్, ఆక్సిజన్, సల్ఫర్. These are the different things which are generally making up of the biomolecules. And we can see that the generally we are using different kind of chemical natures by which we can find out the presence of these biomolecules. And biomolecules levels వచ్చేసి దీన్ని మనం macro biomolecules కూడా అనొచ్చు, because of their size comparatively which is larger when compared to other kind of molecules. Amino acids, proteins, lipids, carbohydrates, and the nucleic acid. ఇంకోటి వచ్చేసి నెక్స్ట్ వన్ ప్రైమరీ అండ్ సెకండరీ మెటబలైట్స్. ఇక్కడ క్లియర్గా గుర్తుపెట్టుకోవాల్సింది, ప్రైమరీ మెటబలైట్స్ ఆర్ ద వన్ విచ్ ఆర్ జనరల్లీ ఇన్వాల్వ్డ్ ఇన్ పర్ఫార్మింగ్ ఆల్ ద ఫిజియోలాజికల్ ఫంక్షన్స్. సెకండరీ మెటబలైట్స్ ఆర్ ద వన్ విచ్ ఆర్ జనరల్లీ we can say that they are the one which are involved in the process of that is they are generally involved in the process of defense mechanism, and also that is generally drugs, defense, and also we can see they can be used, means they have the least function apart from the defense and showing some pigmentation, so they can be used as a drug for the cancer itself. Here we can see macro biomolecules are the one which are generally having different, they are the one which are performing the percentage of total cellular mass, that is the carbohydrates are the highest and ions are the least ones. And coming to the proteins are the one which are generally showing the functions. We can see the proteins The polymeric forms are the one which are generally consisting of peptide bonds, and we can see the peptide bonds are generally created between the amino acids, and the proteins are the polymeric form of the amino acids. And coming to polysaccharides, we can see the polymer of sugars or the nothing but the saccharides, where we can see multiple molecules are joined by means of glycosidic bond, which are involved in joining the monomeric form of sugars to form the polymeric form. And coming to nucleic acids, the nucleic acids are of two different types, that is the DNA and RNA form. That is the genetic material which is generally involved in the storage and the transmission process. And we can see they are the polymers which are consisting of monomeric form of nucleotide joined together by means of phosphodiester bonds. And coming to the structure of the protein is generally made up of amino acids, generally bound by means of peptide linkage. We can see the primary structure, secondary structure, tertiary, and the quaternary structure based on the ability of protein doing the complexity in the function. And we can see the primary is the one which is a native form of the protein, where it is generally folded to form the secondary, tertiary, and the quaternary function. And coming to the nature of bond, that is generally created between the monomeric form, that is the polymeric form of the molecule, where we can see the phosphodiester linkage is the one which is created between the monomeric form of nucleotides. In case of DNA, we can see one strand will be anti-parallel to the other strand, and in between the nitrogen bases they are the hydrogen bonds which are generally involved in this process. And we can see the dynamic state of the body, that is generally the concept of metabolism, where we can see the enzymes are generally undergoing the process of constantly they are undergoing the process of making and breaking. So we can see the turnover is the factor which is generally involved in the metabolism of the enzymes. And we can see the metabolic basis for the living reaction, where the metabolism is the reaction which is generally showing the catabolic and the anabolic reactions together. And finally, what is the living? The living state is nothing but it's always a dynamic state, and it is a non-equilibrium steady state to perform the function and the work, where we can see always the biomolecules or the biomolecules or the chemical components, always they are maintained in a steady state of equilibrium. That is a chemical range where we can see the glucose concentration and the hormones are generally maintained a steady state in the body. So this is about the concept and the summary of this particular chapter, that is 10th of the biomolecules of the botany 1. So meeku elanti doubts unte you can write down in the comment box, and also you can discuss, you can like the you can even like the video and share and subscribe with your friends. So meeku emana doubts unte kuda you can write in the comment box. Thank you for watching.